button. Fantastic. Welcome everyone to today's briefing on CAR T and other immunogeneticities. Very happy to be collaborating with the House Cancer Caucus and extremely pleased to represent or to introduce Representative Derek Kilmer, who represents Washington State's 6th Congressional District. He also serves on the Appropriations Committee. And one of my favorite facts about him is he is an avid Star Wars fan, as am I. So we have that much in common. And without further ado, I'm going to hand over the virtual mic to Representative Kilmer.
Awesome. Thanks, Margarita. And good afternoon, everybody. As Margarita mentioned, I proudly represent Washington's 6th Congressional District, which is Tacoma, Washington, and everything west to the ocean. I also serve as co-chair of the House Cancer Caucus, and it's a real pleasure to welcome you to the first Cancer Caucus event of the 118th Congress. I want to start by thanking my fellow co-chairs, Representatives Brian Fitzpatrick and Brian Higgins, and Mike Kelly, and also thanks to the American Society of Gene and Cell Therapy for being here today and for their continued partnership with our caucus. Most of all, thanks to our speakers today, Drs. Hans-Peter Kiem, Rain Roos, Rainier Brenchens, January McKee, and Marco Davila for sharing their time and expertise with us today. As many of you know, the Cancer Caucus is a bipartisan group of legislators that are dedicated to supporting cancer research and to improving access to cancer prevention and screening, diagnosis and treatment. We're also dedicated to supporting the needs of the cancer community. So if you're a member of Congress or a staff member whose office is not already a part of the Cancer Caucus, please contact my office or the office of one of my fellow co-chairs so that you can join. And don't worry, if you join, I promise we'll only email you twice a day. Now, the work of the caucus hits pretty close to home for me. I lost 3 of my 4 grandparents to cancer and feel lucky that other members of my family are still living due to advancement in cancer detection and treatment. Sadly, we lost a member of my staff, a young woman in her 20s, just a couple years ago to cancer. And I think about her and I think about her family nearly every day. Her family, my family, families across our country aren't alone. Cancer touches nearly every person in this country. This year alone, the American Cancer Society projects that 1.9 million Americans will be diagnosed with cancer and that over 600,000 cancer patients will die. And that's why the House Cancer Caucus and I are committed to supporting robust funding for cancer research at the National Cancer Institute at cancer centers around the country and elsewhere. It's also why we're committed to working with the White House's Cancer Moonshot Coalition to achieve the president's goal of reducing cancer deaths by 50% over the next quarter century. It's also why we're so excited to host this briefing on CAR T cell therapy and other immunotherapies. These individualized targeted therapies reflect the successes of sustained federal investment in cancer research, and they provide new hope for cancer patients and their families. So thank you, and I hope you're all looking forward to hearing from today's briefers as much as I am. So let's get to it. Dr. Kiem, the floor is yours.
Well, thank you so much. And yeah, hello. So my name is Hans-Peter Kiem. I'm the president of the American Society of Gene and Cell Therapy and also professor of medicine and pathology here at the Fred Hutchinson Cancer Center and the University of Washington. And as you may know, members of the American Society of Gene and Cell Therapy have actually developed and provided the foundation for these next generation engineered immune cell therapies for cancer that we will be talking about. So what is gene therapy? Let's see, make sure that we have the slides. On this slide, you can see just a brief depiction of what gene therapy is. It's when we deliver genetic material, DNA or RNA, to cells either outside the body after we collect the cells, which is then called ex vivo gene therapy, or we deliver the genetic material directly inside the body, what we then call in vivo gene therapy. Now, instead of introducing a piece of genetic material, we can also directly edit and make specific changes in the DNA within the cell. And we call this then gene editing, which is more precise than delivering a piece of DNA with a viral vector. So next slide. So how do we make these chimeric antigen receptor CAR cells to treat cancer? The basics are shown here, where we collect blood cells from patients, we bring them to a laboratory and then insert the chimeric antigen receptor construct into a subset of these blood cells that we call T cells. And this will then allow these T cells to very specifically recognize a cancer cell and kill the cancer cell. We then grow millions of these cells and infuse them back into the patient. For example, we can target CD19 or CD20, you may have heard of, which is expressed on leukemia and lymphoma cells. We can do this with very high specificity and kill those cancer cells. So this is really very potent precision medicine or oncology. Next slide. So a bit more how we actually insert the CAR construct, this chimeric antigen construct, into the cell. Now, in most cases, we use a viral vector to deliver a piece of this DNA that encodes for this chimeric antigen receptor on the cell surface. Now, with these viral vectors, all viral genes have been removed. So it's really like a Trojan horse that we use to bring this piece of DNA or RNA into the cells. Now, instead of, you can advance just the rest of the slide, just the entire slide. So instead of viral vectors, we can also use nanoparticles, like lipid nanoparticles, and those are the same lipid nanoparticles that you are probably familiar with that we also use for COVID vaccines. And we can use editing technologies to further improve the potency of these CAR T cells. So we can really make very powerful cells to kill and target cancer cells. Next slide. Now, in terms of the gene therapy pipeline, you can see here most cell and gene therapies in the clinic and being developed now are targeting rare diseases and cancer. And many of the rare diseases are actually cancers like multiple myeloma or lymphoma. Next slide. Now here you can see these are the 6 approved CAR T cell therapies now that we have, and you will hear in the subsequent talks, you will hear a lot more about these different CAR T cell therapies and the success of these CAR T cell therapies for lymphomas, leukemias, and multiple myeloma. And you will also hear about the plans to expand these really promising immunotherapies to solid tumors. And then also have discussions about how we can improve the overall accessibility for these next generation cancer therapies. Thank you. I think we'll probably move straight into the next talk, right?
Absolutely. So thanks, Hans-Peter, for giving that great setup. I was asked to discuss what some of the obstacles are to expand CAR T cell therapy in the context of solid tumor malignancies. Next slide. So again, this is the schematic that was shown. We can create a gene that expresses a chimeric antigen receptor, which is kind of a Frankenstein-like molecule that when expressed by T cells, allow T cells that would otherwise ignore the surface proteins on a cancer cell would now be able to recognize that cancer cell and kill it. And that was the basic foundation when we started working on this over 20 years ago on this technology. Next slide. And this is simply one of the outcomes that prompted the FDA to approve this type of advanced technology. These are the survival curves you can see in children with acute lymphoblastic leukemia that would have otherwise extremely poor prognoses. And you can see here that a significant portion of these patients persist and survive for prolonged periods of time. Now, fortunately, similar studies have been published by our group and others demonstrating the same to be true in adults with acute lymphoblastic leukemia that have about a 3 to 5% long-term survival if the disease initially comes back. And other blood cancers such as lymphomas and multiple myeloma have also led to approvals of this technology, which makes it very promising in blood cancers. But as Hans-Peter pointed out, these are relatively rare cancers, and the much more common cancers are solid tumor malignancies. Next slide. Colon cancers, breast cancer, prostate cancer, lung cancer, and so on. And this is just a cartoon to illustrate that not all cancers are created equal, which is to say that the challenges that we were able to meet to get this technology to work quite well in the context of blood cancers is probably not going to be sufficient in the context of solid tumors, because if you look at the solid tumor cells, the ones in purple, they tend to scaffold themselves with other types of immune cells that would suppress the CAR T cells. They're designed to do that. And that would include things such as fibroblasts and macrophages and a subset of T cells that can inhibit their counterpart T cells that are designed to kill the tumor. And so this means that there are a lot of obstacles that we still need to overcome, and we need to do research in to allow this proof of principle that we have demonstrated in blood cancers to work in more common cancers, such as solid tumor malignancies. Next slide. And so what we and others have been working on over the last 10, 15 years is designing this next generation of CAR T cells. We coined the term armored CAR T cells because for us, an armored CAR is one that is additionally genetically engineered to stimulate, to protect these T cells from being suppressed once they reach the tumor, but in addition to be able to stimulate the immune system that already exists in the patient. And there are a variety of ways you can do that. But one of the ways that we looked at is engineering the T cell to secrete proteins that we call cytokines that can stimulate not only the T cells, but also other immune components. Next slide. And this is just one quick snapshot of a mouse experiment that we did, where we did just that. We took the cytokine IL-18 and put that into a CAR T cell that recognizes CD19. And as you can see here, when you look at just the CAR T cell in red, there's no survival benefit. A T cell that just secretes IL-18, there's no survival benefit. But when you put the IL-18 into the CAR T cell, so it's designed to secrete it at the site of the tumor, all at once you get 100% long-term survival benefit. And this is just one of many examples where where investigators are additionally engineering these CAR T cells to be able to overcome these obstacles which are present in the context of solid tumors. Next slide. Now, I would be remiss to say, I would say that there are scientific obstacles, but there are also political obstacles. So this is a great forum to point this out. As we develop new CAR T cells, the biggest chasm that we have to moving this into the clinic is largely financing. It costs about $3 million to conduct the first trial in humans with any new CAR T cell design. And it is very difficult in this current era, where are you going to get that amount of money? While we get a lot of great funding to do a lot of the mouse experiments from the NIH and the NCI, the grant funding, to write a grant for that amount is extremely difficult, and they're not offered routinely, just given the sheer cost of doing these trials. But there are other mechanisms of funding which we have utilized and others have utilized. One of which is to get philanthropic funds. The second of which is to get institutional support, which again places the burden on the institution, obviously. And the third is to actually license the technology, which is most commonly done to pharmaceutical companies and biotech. Now, that is a great advantage. It allowed us to treat many, many patients on our CAR T cell trials. The unfortunate thing when licensing to industry is that the scientist, the inventor, loses a significant amount of control over their own technology. And so with that, I will wrap up. So again, scientific obstacles. It's great that we have federal funding to be able to investigate new ways of overcoming those obstacles, predominantly in the context of mice models. Second obstacle, funding to get these novel designs into patients to verify that their efficacy is increased. And I would say that is certainly funding is a significant barrier to this technology moving forward. And I think that that's my last slide.
Fantastic. Thank you, Dr. Brent Jens. And next up, we have January McKee.
Hi, hello everyone. My name is January McKee and I lead the process science group here at Castle Creek. I'm grateful for the opportunity to walk you through the challenges of the CAR T-cell manufacturing therapy. I am a believer for everyone to enjoy the benefits of scientific progress and its applications. So it is my duty not to only discuss the roadblocks, but what we can do to alleviate the pain points. Next slide, please. Dr. Kim already explained and showed in detail the process flow diagram of CAR T-cell manufacturing. Here, I want to highlight the hurdles along the manufacturing process. The first one is obviously the difficulty to forecast, as each lot is custom made for one patient. So one lot is one patient. And this will impact availability of clean rooms, trained operators, raw materials, and complex reagents with sometimes very short shelf life. Several of these reagents have lot-to-lot variability and will require robust qualification program before you could use it for manufacturing of the product. The other challenge is obviously the patient variability in the type of the prior treatment that each patient underwent will potentially affect the quality of the starting material, and this will lead to issues in cell growth and scalability. In worst case scenario, for youngest patients like babies and toddlers who have leukemia, for example, procuring enough starting material or blood in this case will not be even feasible due to their low body weight. So once the product is formulated, there's another obstacle there or pain point, which is the cryogenic storage. Pending release, you need these specialized storage facilities to maintain the product in optimum condition. And once it's released for shipment, the shipment requires specialized containers with temperature control monitoring, lane mapping for the the best mode of transportation and route, and validation. Next slide, please. So to address the cost of goods and alleviate some of these pain points, alternative approach are already being tested as proof of concept in phase 1 and phase 2 trials. These alternative approaches include the use of normal healthy donor cells or programmable cells in lieu of patient material. So that's the column, the third column there in the table. The allogeneic, we call this the allogeneic approach, will start with healthy materials, which with established characterization parameters should eliminate the variability and the scalability issues. Furthermore, one lot of allogeneic product can dose multiple patients and is available off the shelf when needed. The major bottleneck for current FDA-approved CAR Ts right now is the use of lentivirus, as Dr. Kim alluded, as the delivery mechanism to insert the CAR receptors into the cell. The current viral manufacturing process is inefficient with huge losses during downstream purification. This talk will not focus into this, but this is a critical raw material that is a major driver for the cost of goods for this product. So an alternative approach is the use of non-viral delivery, like as Dr. Kim again alluded, which is the LNPs, or use genome editing tools to introduce the CAR molecule. This will require using nucleic acids such as RNA or DNA, which is a less complex to make compared to the lentivirus. So as a conclusion for this slide, the allogeneic approach has great potential to address a lot of the challenges encountered during the manufacturing. However, data on the response rate, remission rate, and toxicity will determine feasibility of this approach. Next slide, please. So this diagram really kind of like shows you the operational and logistical challenge that we encounter when we make this CAR T product. So it starts at the beginning when the host healthcare provider will determine if the patient is eligible. Once deemed eligible, the provider, the healthcare provider, will coordinate the collection of the starting material with apheresis units or sites that they collect the blood. And when in some cases, patient will need to be rescheduled for collection, as at this point, most of them are very, very sick, and this is not the first line of treatment. And then when cells are then shipped to the site of manufacturing, if all goes well, a product is generated. Otherwise, a second collection of cells will need to be coordinated. Hence, you could see those arrows on the back and forth arrows in order to, if there's not enough T cells that are frozen as backup, the release product at manufacturing site will trigger the host healthcare provider to schedule the infusion date and the shipment. So the chain is very involved with tracking, coordination, scheduling, crosstalk between patient, healthcare provider, and manufacturing site. In short, the only way I could describe this is currently a logistical nightmare. Next slide, please. So for autologous approach, the most sustainable and accessible ways to decentralize the manufacturing. Right now, it's centralized as shown in the previous slide. So we could explore decentralized manufacturing or even point of care manufacturing as a possible mitigation strategy to the current challenges. CEDAR has released a discussion paper last year, Q4 last year, to get feedback on this approach. Having a manufacturing closer to the healthcare provider will alleviate the majority of these challenges. As an example, last year, BioNTech launched a BioNtainer, a modular vaccine clean room to improve vaccine supply in Africa. So that's likely kind of like, hopefully, the future for CAR-Ts too. The use of automated units of operations can perform manufacturing steps in a single unit can also simplify the process. Clean rooms are costly to maintain, and the bigger, the more expensive. For maximized space, nurseries or closed single bioreactors can allow expansion of multiple products in a single clean room. And then QC can also be eventually simplified after trending of historical batches and extend the products after extensive process and product characterization. Last and most important is that as we gain more experience and as the field matures, collaboration and data transparency is important to learn from what's not working and what's not working. So this is my last slide, and I will hand it to Dr. Rouse, who will give us insight on patient experience. Thank you.
Thank you so much, everyone who's presented prior to me. You really have done a great job of simplifying and explaining some of the gains that we've had in the field of CAR T-cell and cell therapy. But I want to take a step back and talk about what the patient experience is like. I am Rain Rouse. I am a pediatric cancer doctor and also an immunotherapy doctor. So I mainly take care of children with relapsed or refractory leukemias and lymphomas. Most of the patients that I take care of are ones who have often been told, and their family's been told, that they don't have any other options for treatment aside from CAR T-cell therapy. So I'm in Houston, Texas, but I take care of patients from all over the country, sometimes from all over the world. Next slide, please. So one thing that is clearly obvious from our presentation today is that CAR T cells for blood cancers truly can work. And again, we're actually able to cure people who previously had a less than 10% chance of living. And we saw that we have 6 FDA-approved CAR T cell therapies for leukemia, lymphomas, myelomas, which is really exciting. And we're working to expand CAR T cells beyond blood cancers. And we showed some of the challenges that we have doing that, but our science is ready to tackle those challenges. We've established CAR T centers of excellence. We have partnered with patient advocacy groups. We, as scientific societies, such as the American Society for Gene and Cell Therapy, have really risen to the challenge to try to make sure we have best practice guidelines. These are really fancy and complicated therapies, but we're able to enhance safety for the patients. We are working on developing shared care models that allow treatment closer to home. Dr. McKee showed us that patients often have to travel to a different site, and I'll talk a bit more about that. Next. But one thing that I really want to point out is that we still are unable to get these therapies to many underrepresented and under-resourced patients. And this is not because they are not eligible or not suitable for the therapy. It's because there are a number of logistical barriers, not just with the CAR T product and making it, but also cost and travel and reimbursement practices that really can limit access. Next. So patient access to CAR T cells truly is an uphill battle, and it's littered with roadblocks they must overcome. Next. We've seen that it's a really complex process. So this is the process of making the CAR T cells, removing the blood from the patient, inserting the gene for the CAR, manufacturing them, cryopreserving them or freezing them, shipping them to the infusion site, infusing them into the patients. And I won't go through that again, but I want to just give you a highlight of what's going on the patient side as these different steps are happening. Next. So while we're preparing to even see if a patient can receive CAR T cells, they have to identify a CAR T treatment site. It may not even be in their same city. Often it's not even in their state. They have to obtain insurance approval, which is a really complex process and can be a rate limiting step. They have to coordinate T cell collection. These are not newly diagnosed patients, typically. These are patients who have refractory cancer, which means that they often will have other comorbidities or illnesses that make it challenging to collect their cells. And then once they undergo this collection process, the cells have to be shipped to the manufacturer. Next. It can take up to 8 weeks and sometimes even longer to make these cells. So next, there actually is what I like to call a balancing act that must occur, which is while the CAR T cells are growing in the lab, most patients don't have the benefit of just not receiving any cancer therapy. So you have this fine balance of trying to give cancer therapies that will control their cancer, but also limit toxicities because you don't want to cause another illness or issue that may make them too ill to ultimately receive a personalized CAR T cell product that's been made specifically for them. And then lastly, next, is the infusion planning. So it's arranging travel to the CAR T site, and most patients have to stay within a certain radius of the treatment center for at least the first 4 weeks after infusion. So imagine if someone lives in Louisiana or lives in Washington and has to travel to another place because they may be getting an investigational CAR T cell that's only available at a certain site. Next. So at the center of all this is obviously the cancer patient. And so the road to, go ahead, next, please. Thank you. So the road to CAR T cells has all of these roadblocks. And unfortunately, these roadblocks disproportionately affect underrepresented, geographically restricted, and lower socioeconomic status patients. So the insurance barriers are great. There are often coverage delays, denials, patients that don't have insurance. And this is despite patients actually meeting the prescribing criteria that has been approved by the FDA. Logistical requirements, again, having to uproot and stay in a different place for 4 weeks, not being able to work during that time period, having to worry about childcare. You can understand how a patient who may be of lower socioeconomic status or not have a lot of support may have delayed consideration even for these therapies. So they're often referred at a later stage. And then unlike personalized targeted drugs, these factors often need to be addressed even prior to referral, so you can understand how there may be some implicit bias that will make a provider who is well-intentioned say maybe CAR T isn't right for you because there's so many hurdles and barriers we have to get through. Next. So the harsh reality is that despite federal guidelines that mandate private and public insurers to cover commercial CAR T cells, like those 6 FDA approved ones, and to cover the routine costs that are associated with an investigational CAR T cell on a clinical trial, there are a number of delays and denials that make this therapy really inaccessible to many. Why? Well, there is variable policy implementation even at the state level. Like I said, I'm in Texas, but I take care of patients from all over the country, and there are different hoops that the providers and the patients must jump through to get approval for these therapies based on what state they're from. There's a lack of enforcement of federal regulations that require insurers to cover these therapies. There's a lack of transparency in coverage requirements, and there are different requirements and different appeals and denials processes depending on where you are. And lastly, a lack of provider time and knowledge on how to navigate these barriers, and certainly a huge barrier to the patients and families to not know how to address an insurance denial. So I really wanted to bring home the patient experience, and I believe that's my last slide, and I will hand it over.
Thank you, Dr. Rouse. And next up, we have Dr. Marco Davila.
Thank you. So my name is Marco Davila. I am the Senior Vice President of Translational Research and Vice Chair of Cellular Therapies at the Roswell Park Cancer Center in Buffalo, New York. Next slide. So I have the task to talk about everything else outside of CAR T cells, which is obviously a very difficult task. So I'm just going to provide some highlights on really why the field of cancer immunology is so exciting. So as my colleagues have talked about, the cell-based therapies that have been improved are all focused on CAR T cells, where you take an alpha beta T cell and use viral transduction technologies to be able to target these the patient's T cells against their leukemias or their lymphomas. So we are just now scratching the surface for these types of gene engineered cells. So major questions that are being addressed both in academic and industry research laboratories are what other immune cell types may be able to provide us great outcomes for patients, not just with hematologic malignancies, but solid tumor malignancies. So things such as NK cells, gamma delta T cells, regulatory T cells are all being evaluated to determine if they can potentially improve on the outcomes that are seen with alpha beta T cells, but potentially with better toxicity profiles. So immune cell types are a major area of investigation. And here under this other kind of bullet, these CRISPR engineered T cells. So we're also looking at different systems for gene engineering. The first ones, as said in prior discussions, have really focused on gamma retroviral or lentiviral modifications of T cells. So that has been beneficial because the last 20 years of this field has really focused on how can we quickly and easily make viruses, and how can we use these viruses to modify T cells. So we're building on a large body of prior research that's made this process much easier. But there's new technologies that really kind of create new opportunities for gene engineering. So the genome editing technologies such as CRISPR, TALEN, zinc finger nuclease, Caucus, all allow different opportunities. So what's exciting about some of these is they allow us to do multiple edits to an immune cell. Viral transduction, usually we can get 2 or 3 genes in at most. CRISPR genome editing or similar genome editing technologies allows us to be able to do multiple knockouts and knock-ins of these genes to be able to create a cell that potentially can counter some of the suppressive effects of the tumor microenvironment. So those are cell-based therapies, and I won't kind of belabor this since it was discussed before, is also the donor of the cells is something that's an active area of investigation. Autologous, meaning it comes from the patients, versus allogeneic, it comes from an unrelated or a related donor. There's benefits to that because it allows us obviously more rapid infusion of these cells as opposed to collecting, producing, and creating a 4 to 8 week gap where patients have to wait until these cells are ready. Other forms of immune cell therapies. So one of the first cell therapies ever to be improved for patients with cancer was a dendritic cell-based vaccine for prostate cancer. And this is something that other cancer vaccines have mostly fallen out of fashion because of the difficulty of commercializing that product. but with new gene engineering technologies, with other immune cell types, this is now really becoming an important part of new investigations to be able to see if we can utilize cells to be able to stimulate a patient's immune response against their solid tumor malignancy. And probably the biggest, so if we think about CAR T cells that have been approved for, and they're allowing the cure of patients with prior incurable cancers, the partner to this has also been checkpoint inhibitors. So these are proteins that have been utilized, and there's 7 main classes of PD-1, PD-L1 inhibitors that have been approved for something like greater than 90 different indications for solid tumor malignancies. And this is allowing patients with prior incurable solid tumor malignancies to now be cured of their disease. So we have focused mostly on the PD-1, PD-L1 axis, or the CTLA-4 axis, but there's multiple different activation and suppressor axes between a T cell and a tumor cell or the tumor microenvironment that have been reported on, and we're looking the field to determine what other axes can be manipulated to result in better anti-tumor responses. And in terms of other proteins, so antibodies, when I was in graduate school, rituximab was being developed and evaluated for the treatment of lymphoma. It by itself was not active, but in combination with chemotherapy has resulted in the cure of thousands and thousands of patients the last couple of decades. And while regular antibodies have been limited, have not necessarily been able to recapitulate that success of rituximab, what people have realized in this protein engineering field is you can use antibodies to essentially be delivery agents of toxins to tumor cells. So these antibody drug conjugates are very exciting and getting new approvals for patients with leukemias and lymphomas, and we anticipate potentially solid tumor malignancies as well. The bispecific antibody is now taking that protein, that antibody concept on its head, and instead of the antibody meeting destruction of the tumor cell or delivering a toxic agent, what it does is bring a tumor cell in close proximity to a T cell and immune cell, and allows that T cell then to get activated and kill the tumor cell. So there's at least one, just recently, a second approval of a bispecific T cell engager in this class. So another exciting type of product. And one thing that we haven't discussed a lot is cytokines. So when I first became interested in cancer immunotherapy a couple of decades ago, a lot of the clinical experience at that time had been focused on cytokines. This idea of putting these cytokines in because they're going to be able to activate the patient's immune system and be able to then mediate an anti-cancer response. But the problem is that these cytokines are usually very toxic. However, new protein engineering and gene engineering techniques are creating essentially novel receptors that can really only be identified by these kind of synthetic cytokines that may allow the administration of these cytokines to be able to mediate an anti-tumor immune response, but without the toxic complications. So we've really just scratched the surface, and this has been a couple of decades worth of research that has generated amazing outcomes for patients the last several years. And when I say amazing outcomes, I mean cures. And the hope is that with these new pathways of research, we're going to be able to close that gap between the patients that aren't responding to these therapies to potentially generate new responses. And with that, I'll say thank you.
Fantastic. Thank you, Dr. Davila, and thank you to all of the presenters today. So we are going to open up for questions from the participants. If you have a question, if you can use the raise your hand feature. And before we get into the Q&A, I want to say a huge thank you to the staff of the Caucus co-chairs, Representative Kilmer, Fitzpatrick, Kelly, and Higgins. The staff has been amazing in collaborating with ASGCT to put together this briefing. And so I see Sam and Jessica have their hands up, so I will let you guys duke it out and see who wants to go first with the question.
Sam, I think you had your hand up slightly before I did, so you get to go first.
Okay, I'll jump in. First, let me echo Margarita's thanks to all the panelists. This is a wonderful briefing, and I think it's among some of the most important research funding that Congress does every year is innovative things in the oncology space. I want to ask quickly, this is really a question for Ms. Rouse, but I'll also open it up to the other panelists, because I think it kind of touches on categories that each of you mentioned. But Ms. Rouse, you touched a little bit on payment issues, particularly in the commercial insurance space. Where do we stand with payment issues in the Medicare space and with CMS? I mean, one of the things we know is that CMS and Medicare often set the standard that commercial insurers follow when they're considering payments. And we also know, I mean, Ms. McKee touched pretty extensively on sort of the value chain of bringing a product to market. We know Medicare doesn't always value sort of downstream things as effectively as they do actually treatment delivery. So as we think about this from a congressional perspective, how should we be talking to CMS? How should we be encouraging them to value CAR T cell therapies and other therapies in this space to make sure that patients have the best access?
Absolutely. That's an excellent question and such an important one. So CMS and Medicare and Medicaid have actually taken the lead in working with these companies that have commercialized these CAR T cell products and actually have put out very clear guidelines about coverage and reimbursement. One of the biggest issues is that we see is that despite these guidelines, there's not a lot of follow-up and there's not a lot of enforcement. So the issue is that, especially on a state-by-state level, even for CMS, they are able to place a different additional restrictions. They're able to ask for additional information that is outside the actual label and prescribing criteria. And what we're seeing more often now than denials is delays. And every single person that takes care of patients on here will tell you a delay can be just as detrimental to a cancer patient, especially one with relapsed or refractory disease, as a denial. So what we're seeing is that there's a lack of enforcement and follow through. So what we really need to happen is we need policy change to really ensure that the guidelines are being enforced. I have a number of examples, and we all do, and we're all working together to try to really push these insurance companies to actually follow the guidelines that have been laid out. On a private insurer standpoint, you can imagine that there's even more variability and leeway in what they will actually approve. So the big thing is, you've created these guidelines, now let's get them enforced. And it really needs to be, also we need to work on transparency. We need to be sure that it is transparently available what the appeals process is, why are these additional requirements in place, what the timeline to approval is. When something is urgent, it should be within 72 hours. It often will take over a month to get insurance approval for these therapies, during which time the patient will often become ineligible. Thank you.
That's a great point. And I would just say this as well. The labels are very, very minimal for approval. Diagnosis, lines of therapy. So when a payer says you need to get not an echo, but a MUGA, the question is why?
Why?
What if the number shows 35%? Are you going to say no? Then show me the data. So it's a delay game, which is since we're already juggling a lot of issues, the delay just really makes it harder. And I'm sure there's patients that we're not able to treat because of that delay. So great point, Rain.
Yeah, I
Thank you
totally
both.
agree.
Thank you both. You raised excellent points. Jessica, I see your hand is up next.
Great, thanks, Margarita. And I will also just echo everybody else's thanks to the panelists for being here today and for everybody else who's joined. I think, my question is sort of primarily for Dr. McKee. I know that you talked obviously a lot about the idea of the decentralization of manufacturing in the CAR T space, or even doing it at the point of care. I think this is something that I've talked to with folks at Roswell Park about in the past, about the ability of, sort of individual cancer centers to actually make CAR T therapies for their patients in-house. And there are all sorts of benefits to doing that. But I wanted to ask about if we have a sense of what the potential sort of FDA approval process for something like that would look like. I think one of the challenges with really, really novel therapies, like what we're looking at now in the cancer space, is that, you know, FDA just does not have prior experience with these types of therapies. And so we're all sort of, you know, going into this uncharted territory together. And so I just wanted to ask about, you know, sort of what you think the FDA process for that would look like, or what some of those hurdles there might be.
Yeah, that's a really difficult question to answer right now, because we have so there's like stem cell transplant has a different pathway. It's regulated under PHS, I think 561, if I'm not mistaken. And because this goes beyond what that regulation falls, which is minimally manipulated, is the language that the guidance or the regulatory health that PHS says, it does not now fall under like bone marrow transplant or stem cell transplant. It falls under a different category, which is then be oversighted by FDA. So there's a lot of push, obviously, to start using like academic excellence, like Roswell Park or other institutes that have this capability to generate CAR-Ts at the point of care. And I think EMA or Europe is also going into the same space with their PRIME program. It's almost like an RMAT designation, but you know, there's it's kind of hard right now, because even with decentralized manufacturing, FDA is kind of like giving all these like signals that there's you know, right now they're already understaffed with like sites inspection of the manufacturing sites, and in making it decentralized, make it much more sort of less controllable. So I'm not saying that it's not feasible. I think the technology needs to catch up with that. Like I said, you know, there are modular clean rooms now that you could deploy anywhere around the world, and it has its own HVAC system, and even use like automated units that can do from end to end, start to finish of the manufacturing. And some would even have the QC also done on that.
Great, thank you. I see we have a couple of hands up from the audience. So what I will do is I will call on Surya and then Tony. We were going to unmute you, or you'll have to unmute yourself to be able to ask your question. So Surya, you're first. You have speaker privileges, but I believe you need to unmute yourself on your end. I see you took your hand down. Tony, would you like to ask the question?
Hey, this is Tony from Rep. Aragon's office. My question is for Dr. McKee. You mentioned an issue sort of about that. So the viral manufacturing process to create lentiviruses are very important to the creation or transfection of sort of patient T cells to CAR T cells. I'm curious because I remember seeing a Stat News article last year mentioning the sort of lack of availability or limited manufacturing capabilities of getting CAR T cells developed so that patients couldn't get therapies in time. I was sort of curious if you could sort of clarify on how that process looks like, and if there's any recommendations you guys have in terms of how, from a policy standpoint and congressional standpoint, how we could sort of reinvest in the biomanufacturing space back in America. Because my understanding is that a lot of these lentiviruses and reagents that are used for these important therapies are often sort of, developed from, in nations in China and Europe, and depending on what's happening over there, it affects sort of how patients here domestically can access important therapies like CAR T cells and sort of, yeah.
Again, thank you for that. Again, that's a great question. As you have seen with even AAV approved drug therapies, are like up to 3 million, I think right now is the highest so far for gene therapy that is viral based, in this case, AAV. And really, I think it boils down to the technology. Right now, we're kind of like lumping biologics, which are monoclonal antibodies, with advanced therapeutic medicinal products, or like viral vectors, and in this case, CAR-Ts, because it's such a fast-moving pace, and there's not much data right now from a regulatory perspective on how you could, make this product a little bit more. It does not necessarily fit the biologic space because they're bigger. Like the monoclonal are very small in size, while the CAR-Ts obviously are, T cells are like 12 micrometer in size, and viral vectors are like between AAVs are 40 nanometers and lentis are 120 nanometers, 80. So the requirement to do this sterility at the end of the manufacturing of the virus is mostly the reason why we lose like 80 to 90% of the virus during that process. And it's because you have to do sterile filtration, and that's kind of like the regulation right now, that you do need to do sterile filtration at the end. And I understand that you can do that with small molecules and with monoclonal antibodies because they're smaller, but the sheer force of pushing that material, which are slightly bigger than monoclonal antibodies, through sterile filtration, because that is the regulation right now, is just what's causing a lot of these issues with viral manufacturing. There's a lot of failure to generate enough materials to be able to dose to use for CAR-T downstream. And the way that we're approaching right now is trying to close that part, so that if you do close the system, because it's very manual, if you are able to make these units in a single closed system, then you might not need to do sterile filtration. We don't do sterile filtration for CAR-T, obviously. So we do need to demonstrate a way that we prove that the process is aseptic or aseptic throughout. So you just need to demonstrate aseptic process validation, and you show that you do 3 runs or multiple runs using the process in like a water run with broth, and then you demonstrate that there's no growth of that, then you could avoid sterile filtration. So that, again, it boils down to the technology. The upstream part, the making of the virus, is getting better with generating enough virus, but the downstream is the issue right now.
Thank you, Dr. McKee. I see that we are 2 minutes past the hour, and just in the essence of time and trying to be conscientious of everybody's schedules. If you have any questions that were not answered, please feel welcome to email ASGCT staff directly, and we will make sure that we respond to you with the answers to your questions. Or if you have any follow-up requests to meet with any of the panelists, we're happy to facilitate that as well. I'd also like to thank, in addition to the panelists, I'd like to thank their institutions as well. Fred Hutchinson Cancer Research Center, Roswell Park Comprehensive Cancer Center, Castle Creek Biosciences, and Baylor College of Medicine. So in addition to all of the Cancer Caucus co-hosts and our panelists and your institutions, I also want to thank you as attendees for participating in this very hopefully informative briefing. And we hope to keep this conversation going and be a resource to you with any future questions you have on gene and cell therapies. Thank you very much.
Thank you.